So if I stay within my 18 minutes, I will have a visual out of the box. Do I have a pointer in advance, sir?
No, I can.
Okay, all right. When I was asked to do this, I wanted to talk about two of my passions, which I really think fit within the TED program, and that's really genetic disease and children's research. And I'm going to tell you about a devastating children's disease. But also my other passion is education and how we can just really invigorate the youth of today to participate in science, and also to get parents engaged in science, so we can train the next generation of researchers. So if on the next slide. What is genetics? I'm going to talk about genetics. This is that black box term that we read about sometimes, but what does genetics mean? Next, please. So genetics is actually a complicated business. We're in the realm of what we call a revolution in medicine and in research right now. We now have the capability to inquire about the 3.4 billion letters or base pairs that actually encode our genetic code and how they are actually inherited. So next. So let's think about that and just take a moment to think about 3.4 billion letters. So let's just put it into the perspective of some books. That's 200 volumes of the Manhattan telephone book. The other thing is I could stand here and recite the genetic code at a reasonable pace, and it would take me 9.5 years. That's an incredible amount of data that we're now able to inquire in the realms of genetic disease. So next. So just to get some ground rules and some basic words out of the way, DNA is that 3.4 billion letters, and that encodes genes. That's the template. That's really the computer code. And we take that computer code, or a cell takes that computer code to make something called RNA, and that's the message. That's the communication in cells in terms of what does a cell want to do, how is it going to work. And that makes a protein. That's the translated into the protein. And the proteins are the workers in those billions and billions of cells that populate our body. It's estimated there are around about 24,000 genes, and 24,000 proteins are made from those genes. But I just told you there's 3.4 billion base pairs or letters. Only about 1% actually encodes those nuts and bolts, or the proteins that we actually, the worker bees in the cells, which is incredibly complicated for us. That means there's 99% of that code, and we don't understand it, is probably regulating or controlling why we make some of those 24,000. So I'm just going to walk through a couple of things here. What does DNA sequence look like? It's actually only 4 letters. It's an A and a C and a G and a T with multiple combinations. And this would be how it would look like, obviously, in letter form. And this is just a printout we would get if we actually used some molecular biology techniques to sequence some DNA from any one of you right now. That DNA is packaged on something called chromosomes. So these are just teased out from cells, and this is just a schematic for the 23 different types of chromosomes that actually are packaged. So the DNA is all supercoiled in a particular way, and it has all these other complicated things that keep it integral within a cell. So every cell, whether it be an immune cell or whether it be a neuron, or this is just a schematic, pretty much has the same basic engines that drive it. The DNA is in that nucleus, and then we have all sorts of other engine parts that we give complicated names like mitochondria and endoplasmic reticulum that keep all those proteins and the nuts and bolts functional. But what happens when something goes wrong? Well, that's inherited disease. So inherited disease is really where we're trying to study that 3.4 billion base pairs right now in the context of why a child might have a rare disease. And I'm going to talk a little bit about that. But before we get there, just say, okay, what is autosomal recessive disease? Why may a child get something like Batten disease, which I'm going to talk about? Well, out of that 3.4 billion base pairs, it's estimated for those 24,000 genes that any one of us, and this is an estimate now because we really don't know this, is that any one of us may have maybe 10 of those that are broken. But that's okay because we have 2 copies. So when we have a child, we're donating one of those copies of every one of those 24,000 genes to make our offspring. Now, if we have a bad copy of one of those 10, well, that's okay, because your partner may be giving you a functional one that works. Oh, can we go back to the other slide? I'm sorry. And that's pretty much what this is here. So let's assume right now, these 2 individuals, they unfortunately, out of that 24,000, have the same broken copy of the same gene or protein or nut and bolt. So that means, well, if we think about the jumbling up here, there's a 1 in 4 chance that they could get the broken copy from both of their parents. The chances are there's a 2 in 4 chance they'll get 1 broken copy, 1 good copy. or they may just get 2 good copies. 1 in 4 chance is inherited autosomal recessive disease, and it's devastating. These are just some statistics. Batten disease I'm going to talk about. There's about 750 children in the United States right now with this inherited genetic disorder. And I just randomly put some others up from the National Organization of Rare Diseases here. But just to tell you, when you start accumulating those numbers of children, and adults may have some of these rare diseases as well. It just depends on when these diseases start. There's over 4,000 of these rare genetic diseases. So we're totaling up 3 to 4% of our children right now. So individually, many of you would not have heard of Batten disease because there's only 750 children in the United States with it. But when we start adding up the numbers of children, we have a lot of these rare diseases that we need to study. So Batten disease is the one I'm going to give you a little bit of a backdrop on. So this is a cell, very simple schematic of the cell. So what happens in Batten disease? Well, it's just to think of one of those genes that encodes one of those proteins of the workers are these scissors right now. And there's this big thing called the lysosome, which is the garbage disposal in cells. Just like our cells need to eat and they need to break things down. So the lysosome is the garbage disposal of the cells. And what actually happens in Batten disease, if you just hit the key one more time, and then 4 times consecutively, those scissors break and we get garbage accumulating in the lysosome. So that's because this is the recycling organelle of the cell. Just for reference here, this is the nucleus where all the DNA is, which is encoding things that pass around the cells, and some of it goes to the lysosome. So we get accumulation of something in the lysosome, and it's a very complicated business because we don't really know what's actually accumulating in there because we don't know what all the 24,000 nuts and bolts in the cell are supposed to be doing. So what my lab actually studies is what actually happens in an awful case of Batten disease, because one of those nuts and bolts is broken, causing all of that happening. And every time something like this happens in the lysosome, it results in what we call a neurodegenerative disease. So next up. So Batten disease. This is really a very cold description of Batten disease. So what will happen in Batten disease? There's many, many diseases where children are born with what we call congenital defects, which they will die from. Very cruel. This is equally as cruel, but in a different way. A child with Batten disease will be perfectly normal at the age of 5. They will start to lose their vision, probably receive a pair of glasses that will improve their vision. Within 3 months, a trip to the optician will reveal they will need a second prescription because their retina is deteriorating. By the time they are 7 years old, they will probably be legally blind. By 8, they certainly will be. Following this, after learning Braille, because you educate your children, you love your children, you want them to progress, they will lose the ability to read Braille by touch because the disease starts to ravage the brain even more and they degenerate. They lose their motor skills, they lose their ability to speak, they lose their ability to walk. They will end up in a vegetative state anywhere between the age of 15 and 25. It's highly variable for many reasons that we just don't understand. I could have 2 16-year-old girls that I've known in my lifetime with Batten disease. One would be here giggling and having fun with you guys. She'd be blind, but she'd be at the mental state of maybe a 10-year-old. That other 16-year-old would actually be in a wheelchair in a vegetative state. And I can tell you the 16-year-old I'm talking about right now, when she got to 18, she crashed to that vegetative state within 3 months at the age of 18. It's truly a horrible disease. So, something that's important that I study and I have a passion for. Next up, I don't want to leave you with such a miserable story because obviously we have to do something about it. I do try to pluck at heartstrings because I desperately want more people to know about this and to work on it. So how do we go about that? As my fellow compatriots many years ago once said, and now for something completely different. So how to get to Batten disease, and how did I get there, and how would I like more people to get there? Well, science is cool. Science gets a really bad rap. Generally, we're geeks, we're nerds, and we are, but it's a fun thing. We're just as competitive as any athlete that I know. We want to beat things, we want to set records, we want to cure things. Beating Batten disease is like this. Michael Jordan takes that 3-point shot with 1 second on the clock, makes it. Beating Batten disease, being a scientist, is the next time you go to take that three-point shot, someone's moved the hoop up 5 more feet. Because every time you make a discovery, it gets harder and harder. So science shouldn't be scary, or it shouldn't be hard. Science should be taught age appropriately, although at the end, and I'm doing pretty good on time so far, I'll demonstrate something that can be done at any age. So if you just put the on there and click on. So this is a simple experiment. So I've done teaching at all ages, and this is introduced. Oh, go back, and then if you just put the thing on there and click on. There we go. So this is introductory science at kindergarten level. What we do. There's 2 magnets there. You see, you push them apart, and you go, oh man, why is it doing that? Why does it do that? And then when we flip it over, it attracts. Well, there's a different challenge. There's some free electrons floating around in there. So you need to have children to inquire. And of course, it's got to be fun. We can take those magnets and get some iron filings and make a smiley face. Next up, and we might want to move maybe to something to 1st grade or 2nd grade, and this is a fun one. I'm kind of giving it away here because we put what's on there. If we click this one as well, we've got 2 little beakers here with tap water and salt water. You don't tell the kids what it is, or you can actually have them mix up the salt in there and see what happens when an egg, and there's no trickery here, sinks in tap water. But because there's something called sodium chloride in the salt water, there's a change in the density. So why does it do that? The inquiring mind needs to be encouraged. So then if we move on to the next thing, maybe we go 3rd and 4th grade. Who doesn't want to look at tadpoles going into frogs? And all of the questions about biology you get with respect to why the legs are coming and the tail is going, and there's all these complicated biological terms like apoptosis, which you'll learn when you go to college, which actually explains something like that. Okay, and if we move on and we click this one, now this is the one we may or may not have some problems with. It was okay on one computer and not on the other. Oh, and it's not okay now. Okay. But what we want to show here is you just go to the video file. It's a great idea. Thank you.
Which one is this?
You got it, that one, right there. Okay, I'm watching my time penalty here, by the way. There we go. So what we've got here is this is actually a DNA gel electrophoresis. So you can do this. We do this with middle school students right now. And what's happening here is we've loaded DNA and we're just going to separate it on a gel. This is the prerequisite of, okay, we can take a piece of salmon from Hy-Vee or an apple from Hy-Vee, mush it up in a dish and do some extractions and then run out on this gel. Hopefully you'll see the blue line is where the DNA would actually be migrating. And then we'll take a visual in just a minute and show children what DNA looks like before we would do that big genomic sequencing to maybe inquire about that 3.4 billion base pairs. So it's moved. So then if we go back onto the next PowerPoint slide, there's various ways you can see this. So I've just taken some images, and that gel, depending on what we would apply to it, you could look at it this way, or you could just do different types of staining and look at that DNA. This is entirely an age-appropriate activity for middle schoolers, and that really gets the middle schoolers when we lose them. It really is. And not just because we're not good at teaching it, it's because we're getting to that point where as parents we may not have been exposed to something like that. So as parents, it's kind of easier for me with this, but when it comes to other subjects like chemistry and physics, which I was terrible at, as they're taught and those worksheets come home, magnets, I remember magnets, yeah, I know what that's about. Frog life cycle, DNA, I kind of know that a little bit. So we need to educate a little bit there, because when we move to the next thing, we need to do a better job in understanding inherited disease, because we have 3%, maybe 4% of our children right now with these debilitating diseases. And I told you about Batten disease and how horrible that is. Many of them are like that. Children die from these things. We need to do research. We need to find cures. So, science is observation. Biology is a variable. When I'm studying Batten disease, I'm asked, how can I figure out what's going wrong with the brain? We don't understand how the brain works in its entirety right now. It's like if you go take a car to be fixed right now, they plug a computer in, it comes up with that error code. I know what part of the brain starts to degenerate in our models for this disease, and I think in the children right now. So I go to the table of contents, and I flip the pages, and I have blank pages. We've got a long way to understand the human body, and one of the other TED Talks meant that. And I have 3.5 minutes now to show to you why biology is so variable and what an age-appropriate activity actually is. So I'm going to make a mess, if that's okay, but I did bring some. So I do this with every sort of age group, and it kind of helps that you're further back because there's this thing here. So we have these 2 eggs, or these 2 eggs. And actually, so I'm not hiding anything, I'll bring them all out here. We have several eggs. Now I've just juggled those around. So, okay, there's an egg. There's an egg. Just from superficial examination, and I kind of like to mix it up myself. I didn't know which one was going to hit the floor first. One is hard-boiled as one is not hard-boiled. Now when we want to look what's inside here, it's the same thing. You really don't know what's inside there. So the next thing I would do is to have students do this. And they were real ones, but I know I can juggle. You get better when you practice juggling. So, well, let's try 3. So I don't know which ones I have here. Well, juggle. But the big thing is that. You don't know the difference with those things. And the absolute message there is science should be fun, but don't be fooled by biology because we really have a long way to go to understand it right now. Thank you.